Our main objective in this work is to identify the DNA sequence by our novel Z-shaped graphene field effect transistor. Here we can see the chemical structure of DNA. It is made up of nucleotides. Each one of these nucleotides consists of a sugar group, a phosphate group, and a nitrogen base. There are 4 types of DNA bases, which are adenine, thymine, guanine, and cytosine. The DNA sequence, or the genetic code, is determined by the order of the 4 DNA bases. DNA sequencing is the process of reading of the sequence of DNA bases within a DNA molecule. Here we can see a double-stranded DNA. So, why do we need DNA sequencing? What is the importance of DNA sequencing? DNA sequencing helps identify individuals since each individual has a unique genetic sequence. Also, determining gene structure and function. Moreover, DNA sequencing is a critical step to improve human health and to achieve personalized medicine where the right treatment will be given to patients. In our work, we are using quantum-wise atomistic toolkit and virtual nanolab in order to design the Z-shaped field effect transistor and conduct the simulation to get our results and test the sensor. Here we can see the designed sensor. It consists of 2 electrodes, a semiconducting channel, a nanopore in the middle, and a gate. In this case, we are using thymine. This kind of simulation is very accurate and takes a lot of time. So, in order to speed up our process, we are using high-performance computer. In our simulation, we used 7 nodes where each node has 36 processors. In total, we used 252 processors to conduct our simulation. The main idea behind our developed Z-shaped field effect transistor is that the 4 types of DNA bases, adenine, thymine, guanine, and cytosine, have unique electronic densities of states, which are identifiable and can be utilized to distinguish among the 4 types of DNA bases. In order to conduct our simulation, we used first principle modeling. Each one of the DNA bases were placed within the nanopore, and the simulations were conducted for 4 different types of orientation. Also, the translation along the z-axis were considered for each base. Our results indicate that each base will result in a unique electronic signature, which shows very good results for future DNA sequencing. The effect of decorating the sensor with gold or silver nanoparticles was studied, since these nanoparticles has high affinity to DNA bases. It is observed that the bare sensor produces higher electronic current than the sensor decorated with gold or silver nanoparticles. In conclusion, we have theoretically analyzed the Z-shaped sensor's performance for the purpose of DNA sequencing. Future work may include electronic transport calculations for DNA nucleotides in a salt solution.